So our next speaker is Dimitris Sioutopoulos. He works at the Center for Research and Technology, Hellas, in Greece, in the Chemical Process and Energy Resource Institute. And his presentation today is about innovative and sustainable solutions in the dairy industry, which was done in the SPOTVIEW project. Dimitris?
Yes, I'm listening. Dennis?
Yes.
Can you listen okay?
Yes, we hear you loud and clear. Can you switch to the next slide? Yes, exactly.
Okay. Thank you. Good afternoon to everyone. I'm Dimitris Sioutopoulos. In the next few minutes, we're going to talk about some of the results that were obtained, as we said, from the SPOTVIEW project. In collaboration with the dairy industry of MEVGAL. Very briefly, the outline of my presentation. At first, a small introduction about the dairy industry in the European Union and also some information regarding membrane technologies. Then we're going to talk about the results of laboratory tests that were performed in order to recover valuable compounds from the dairy effluents using membrane technology. Based on these results, a pilot unit was designed, constructed, and operated for a period of 3 months in the premises of MEVGAL, the dairy industry. Finally, the conclusions of our study can serve as a point for further discussion. In this diagram to the left, the total milk production for the European Union is shown for the last six years. As you can see, for the year 2014, approximately 150 million tons of milk was produced. The milk production for these 6 years is continuously increased. So in the last year, we have a production of 160 million tons of milk. Regarding the share of each country in the total milk production, as you can see in the pie chart on the right, Germany, France, and United Kingdom are the main milk producers with a share of roughly 20, 15, and 10% of the total milk produced in Europe. We all know the main dairy products, but what is the milk in the European Union used for? According to the data from the Eurostat, the majority of the milk, in a percentage of roughly 38%, is used for the production of cheese. Another 29% is used for butter, 12% for cream, and only 11% is used as drinking milk. Let's move on. Okay, this slide is very important for us. We made some simple calculations and that explain why the SPOTVIEW project is so important. By using the coefficient of specific water consumption for the dairy industry, we were able to calculate the total water consumption for the whole Europe. I must say that this coefficient of 4 cubic meters of water per ton of milk is rather a conservative estimation, and thus that the real number regarding the annual water consumption in the dairy industry is higher than the number you can see. The successful outcome of the SPOTVIEW project through the application of novel technologies will result in a reduction of roughly 25% of water consumption in the dairy industry. This means that we will be able to save approximately 130 million tons of water per year. This amount is quite huge and is enough in order to cover the water needs of a city with the population of Barcelona. So I will talk for a great amount of water that can be saved. Membrane technology is an established technology. It's a physical separation with very low energy cost. There are 4 main types of membranes based on the size of the membrane pores: microfiltration, ultrafiltration, nanofiltration, and reverse osmosis. This membrane is used also in the dairy industry in order to recover milk components such as the fat globulus, the casein micelles, whey proteins, and nanolactose. The most commonly used membrane type in the dairy industry is the ultrafiltration, followed by nanofiltration, reverse osmosis, and microfiltration. Let's move to the experimental results. These are some pictures of the lab scale setups that were used for the recovery of valuable compounds. On the left is a pressurized membrane cell where different membranes were used and examined the efficiency regarding the recovery of valuable compounds. While on the right, you can see a more sophisticated lab unit equipped with a touchscreen that enable us to adjust remotely all the operating parameters. A sample of flashing yogurt and flashing milk were selected as the most appropriate samples for the recovery of valuable compounds. I must say that flashing is a cleaning procedure using potable clean water that is used several times during the day in the dairy industry in order to remove the remaining products from the pipes and the tanks. This is necessary due to the variety of products that are produced during the day in a dairy industry. These are some pictures taken before and after the laboratory test. Since my time is quite limited, I will go to the next slide. These are some preliminary results. I'm going straight to the results of the lab test. And I can see that the retention of value in these diagrams on the left, there is the retention of flashing milk, and on the right, the retention of flashing yogurt regarding valuable compounds such as lactose, proteins. Based on the experimental results, it is concluded that ultrafiltration membranes is the most effective way to remove these valuable compounds. If we would like to remove also the lactose, another type of membrane should be used. This type is nanofiltration. Finally, microfiltration seems to be unsuitable for this kind of separations. Based on the results of this laboratory test, an submerged ultrafiltration pilot unit was designed and constructed. I have to say that another pilot unit, an anaerobic aerobic membrane bioreactor, was also designed and tested in the MEVGAL premises in order to produce biogas and to produce also clean water that can be reused as cooling water in the dairy industry. And given the opportunity, I would like to say that our lab has substantial experience in designing and constructing membrane pilot units. In the table on the right, you can see the most important operating parameters of this pilot unit. This pilot unit was fully automatic, remotely controlled through the computer or a tablet or a smartphone. So we were able to monitor and also to adjust all the parameters from our office, from our lab at CERTH, which is 60 kilometers away from the dairy industry. In addition, the pilot unit had also safety alerts and was sending us warning emails in case of increased pressure values or when we had power failure. These are two pictures of the pilot unit during the operation at the MEVGAL industry. In order to evaluate the performance of the pilot unit, two performance indicators were developed. The first one was the retention of the compounds, and the second one was the transmembrane pressure that is strongly related to undesired membrane phenomena such as fouling. As you can see on the first picture on the right, the retention of proteins and the fat was almost completely, while on the other hand, the stable profile of the transmembrane pressures implies that we have very smooth operation and no severe membrane fouling. In the effort to optimize the pilot unit operation, we study the effect of various parameters such as the operational temperature, the backwash frequency, and the transmembrane and the transmembrane pressure. In this slide, the effect of operating pressure is described in the range of 4 to 40 degrees. As you can see on the left, at 4 degrees of Celsius, the transmembrane pressure profile reveals that the pressure is rapidly increased. This means that we have severe fouling and we had to stop the operation and clean the membranes. On the other side, at 20 degrees of Celsius, the operation was quite smooth. We had some fouling phenomena, but this was moderate. Finally, the optimum temperature for this operation is, it seems that is the temperature of 40 degrees of Celsius. On the other hand, on the right, by comparing the rejection of proteins and fats for the three these cases, it comes out that the rejection of these valuable compounds are not significantly affected by the operating temperature. Thus, it can be concluded that the optimum temperature for this operation is, as I said before, the 40 degrees of Celsius. Finally, I'm going to present the effect of the parameter of permeation rate. Concerning this parameter, as we all know, it is desirable to have as much higher values of permeation rate as possible. This means higher productivity. But as you can see, at the higher value of permeation rate, we had very severe membrane fouling phenomena. So we had to stop the membrane and clean it again. It seems that the values at the range of 6 to 8 liters per square meter per hour are acceptable for this kind of separation. As previous, the retention was not affecting for the permeation rate. So, in conclusion, I would say that we were able to conduct a large number of laboratory tests that revealed that the submerged ultrafiltration membrane system was the most suitable for concentrating the flushing stream and recovery valuable compounds. Concerning the optimum condition for the recovery in the piloted unit study, it was revealed that we should use higher temperature values and permit fluxes in the range of 6 to 8 liters per square meter per hour. Thank you.
Thank you very much, Dimitris. As we are already 3:15, there's not much time for questions in the open chat. Well, I think this is a comment, not really a question. I would be interested in the thing that you said in the beginning. You said you aim for 25% of reduction in water use. What did you achieve in the end with
the results were quite promising. This technology I described was only for the recovery of valuable compounds. The the anaerobic aerobic membrane reactor that was also tested in pilot unit scale revealed and that were able to recover more than 25% of water, and that was quite promising. So I could say that the results were successful of our study.
Yeah. Okay. Great. Great. Interesting. So you achieved this. Thank you again, Dimitris.
Thank you, Dennis.